Now, if you were a bacterium, then remember your DNA would be just this giant convoluted loop right in the middle of the cell, and you'd have a bunch of ribosomes floating all around the DNA. And so as soon as you started making that messenger RNA transcript, right, so there's our messenger RNA, you could actually get this ribosome right onto it and start translation, the making of the protein, right away. And that messenger RNA does not last real long. So if you are a eukaryote, then you are going to do something else after making the messenger RNA, and that's the post-transcription processing. Basically what we're going to do is this. First, we're going to cut out these strips called introns, just little segments. We're going to remove them and leave them in the nucleus. Two, we're going to add a couple of stabilizing elements. So one is a cap at the 5' end, and then lastly we're going to add a tail at the 3' end. And all of these lead to stability so that the mRNA can stick around longer and make proteins continuously. So let's take a look at this. We got our DNA, right? It's represented as double-stranded, but it's shown here as these kind of banded segments, and each one represents a different functional aspect, like the promoter we've talked about before, of the DNA double helix. There's also portions called exons and introns. So exons you can think of as the parts that exit the nucleus, and introns, they stay in. Well, all the DNA stays in. So after transcription, we have a transcript of the exons, and we have a transcript of the introns. So enzymes are going to come along here, cut these out, stitch the two ends together, and then all these introns, again, stay in the nucleus, but our exons, all stitched together, end up exiting. The cap and the tail, well, so there's a guanine and this triple phosphate attached to the 5' end, and then a whole bunch of adenines that are added to the other end called the poly-A tail. And these mainly help with stability, and now I kind of think of this as adding sort of like a cap, the little tassel, and this is the gown. You're graduated, you're ready to go out into the cytoplasm and make a protein. Let's pick up where we left off last time with transcription and look at post-transcription processing using the paper models.
RNA polymerase is going to fall off, so this is what's known as termination, obviously. This can separate, covalent bonds are formed between all the nucleotides here, and we have our messenger RNA. Now if you're a prokaryote, before you even finished making your messenger RNA, a ribosome probably would have come out here and started making the protein, doing translation. But if you're a eukaryote, before the messenger RNA leaves the nucleus, and I'm going to set the DNA aside, it needs to be processed a little bit. And there's a couple weird things that happen. One is this little piece that I've grayed out is called an intron. Actually, let me bring this back over here. And this is non-coding information. So one thing that happens is enzymes come and cut that out and stitch back together the rest of this. We don't need the DNA. But these three are just gone, and the rest of it gets stitched together. And there's several purposes for that. I won't get into it right now, but that's one bit that happens. The other one is that they get this poly-A tail and a 5' cap. So the cap is the guanine that goes here and adds stability so this doesn't get broken down. And the poly-A tail is an extension of a bunch of adenines that get added on to the 3' end. So now this has been processed, it's stable, and it's ready to leave the nucleus.